Hello first year students, we are going to see spore staining technique. Now here we are going to take a slide, very clean heated slide in which there is nothing like dust or something. This slide is then after used for marking the smear from the lower part. So in the lower part, with the help of glass marker pen, we will make a long oval shape like this. Okay, so now I think it is very clear to you that how we are going to mark the smear. Within this area we are going to put our suspension of bacteria. Okay, now we will use the nichrome wire which is already heated by us. This is our bacterial suspension in which there will be spores as well as vegetative cells of bacillus species. So we will mix it properly like this, and now you can see very turbid suspension which indicates the presence of spores and bacteria. Now we are going to use this nichrome wire, okay, opening the mouth of the tube, sterilizing the tube, then dipping the nichrome wire inside the suspension tube, again sterilizing the mouth of the suspension tube, put it away, and now we will take the slide, and on the slide with the help of nichrome wire we are going to make the smear. Okay, so this is the way how we are going to prepare the smear. Okay, so here you can see smear is nicely prepared. Now again I will sterilize my nichrome wire, and now I will allow this smear to air dry. Air drying in the bacterial smear is very much required, because otherwise it will distort the shapes of the bacterial cells when we will apply heat. So it is allowed to air dry completely before we go for heat fixation. Okay, if the smear is very thick, it will take more time to become air dried. Okay, so now let us wait for air drying of the smear. Now by the time I will tell you that this is a special technique of staining in which we are going to use different you can say staining solutions. First of all, we are going to see that we are going to use a heavily concentrated solution of green dye, that is malachite green. You can see here very nicely, this is malachite green. Okay, can you read it properly? Okay, so this is our malachite green, 5% malachite green, heavily concentrated solution which will allow by the spore to get inside. Okay, and then after we are going to give the heat treatment also, and after that we are going to use this aqueous safranin solution, that is 0.5% aqueous safranin. Okay, so with the help of these two dyes you will see today that how we are going to prepare the spore staining slide. Now it is already air dried, you can see very nicely, and now I will pass this slide through the burner for 20 times. Okay, so 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18, 19 and 20. So now our slide is already heat fixed. You can see very nicely that slightly turbid area you can observe in the oval shape which you have already made. Okay, so now I will take the tripod, I will keep it on the burner, and before that I will keep a wire gauze. This is the wire gauze, you can see very nicely. Okay, so we are going to keep it on the tripod. Now after that I will keep my slide on that, and Now I will flood this slide with the malachite green. Okay, you can see very nicely that how I'm going to do that. Okay, so I'm going to flood the slide with malachite green. If there will be any problem for opening of the bottle or taking the solution out, you can also open the bottle, there is no problem. Okay, so by opening the bottle you can get the steam, because this remain unused and you are away, so it will be very tight. So now I will pour the solution of malachite green over the slide. You can see very nicely the slide is completely flooded, and now I will give the heat from the below. Now heat should be in the such a way that it should be moderately given, but it should be continuously given till the steam arises. Okay, fine. So now you have to first of all decide the orientation of the burner. Okay, so you just decide that in which area or in which directions you want to move your this burner. Okay, accordingly you will adjust the slide. Now I already adjust the slide. You can see that from the below I am moving the burner, so thereby entire smear will get the heat properly. Okay, so this is the way how we are going to give the heat treatment. Okay, heavily concentrated solution and it is heated, so by both the ways the spores will get stained, because their pores will get open because of both the treatments. Okay, heating as well as the concentrated solution of the dye. Okay, so heating should be done till the steam arises. Okay, so it will take your roughly some minutes. Now if you see that your this staining solution is getting evaporated, then again you have to flood the slide with the malachite green solution. It should not be completely evaporated. Okay, otherwise you will get the precipitates of the dye molecules and your staining will be failure. Okay, so as soon as you see the evaporation, okay, fast evaporation or precipitation, the dye molecules should not be allowed to do this. Okay, so again you can add the solution and you can continue heating, okay, properly from the below. You have to move the burner in backward and forward direction. You can see very nicely. Okay, here we are actually heating the smear, okay, along with the malachite green stain. Now it is to be done till the steam rises. Now I think you are able to see that steam has already been rising, because you can see the bubbling formation in this. Okay, so now here we are going to stop. Okay, allow it to cool completely, and by the time you can off the burner. Okay, now will not require the burner. Allow it to cool completely.